Our next presenter is Richard Morris. He's the CEO of PharmaSeq, and he has a BS from Cornell University and a PhD in biochemistry from the University of California. So with that, Richard, welcome.
Good evening, everyone. Thank you very much for inviting and giving PharmaSeq the opportunity to speak this evening. Okay. Our moonshot is to create a tool that will allow the world to benefit from all of the advances in genetics and molecular biology that have occurred over the last 40 years. The implications of what we are doing, we believe, are extremely profound. Everybody has probably heard that it's relatively easy and inexpensive to sequence, or what we say, read DNA. The price of doing a human genome is now down to about $1,000 per person. This is going to create a lot of opportunities in diagnostics, therapeutics, and things like that. However, less attention has been paid to lowering the cost of synthesizing DNA or writing DNA, that is, creating DNA de novo. The cost of doing this has actually been stuck for the last few years, and it's not following the typical Moore's law type of behavior that has occurred with DNA sequencing. We've come up with a design that we are confident will allow us to rapidly and inexpensively synthesize large libraries of DNA. It takes advantage of inventions that I'm going to describe to you tonight, plus the enormous increase in computing power that has occurred in the last few years. Just as a reminder for those of you that are less biologically inclined, the human genome has about 3 billion individual letters or base pairs. So in this room, there are probably about 1 trillion letters. And it took 12 years to sequence, to do the initial sequence. Not much later, the mouse genome, which is about the same size as the human genome, took less than a week. So that gives you an idea of the pace of technology. So to provide the similar economies of scale that are used in DNA sequencing for DNA synthesis, we're taking advantage of an amazing device which has been invented here in New Jersey called a P-chip. And they're very small. I brought them with me to show people if they are interested. The P-chips are extremely small, and what they've done at the University of Bristol is they've actually glued one onto every single ant in a hive. And they can watch the social behavior of ants and follow their behavior with this P-chip. Okay, so as they go in and out of hive, they can stress the hives and do all sorts of things like that. It's the first time in history that teaching outside of the human race has been demonstrated between two ants. They call it tandem running, where if you destroy their nest and you give them a good nest and a bad nest, they eventually figure out which the good nest is by teaching each other by rubbing their antennae together. And using the P-chips, we're able to track and trace every ant as it goes in and out of the hive. The P-chip itself, and I'll show one here, is a semiconductor device. They're made in the same, oops, sorry about that. They're made in the same foundries that Apple and Dell use. The dimension is 0.5 millimeter. These are the large ones. And it's resistant to just about any kind of a challenge. You can freeze them in liquid nitrogen. You can autoclave them thousands of times. You can heat them up to 500 degrees on a soldering iron, and they will still continue to function. They're basically like little rocks. The way it works is that when the P-chip is illuminated with a laser beam, it transmits a unique serial number which is stored on the chip. Okay? No two have the same ID number. We control that. We also know how to attach chemicals to the surface of this chip. And on it, we can do things like grow synthetic strands of DNA. The P-chip itself is great to tag and track things or to see if something's authentic. They have great use in the pharmaceutical industry to prevent counterfeiting. And we have lots of projects with them. We use them to inject them into mice, like vehicle ID numbers for them. We embed them in plastic or glass. We can attach it to just about anything that's living or not. Besides the ants, they've been used for pioneering studies in sudden hive collapse syndrome. There's some very nice video clips available at the University of Illinois website. So the P-chip itself integrates a lot of the latest technology around. They're completely different than other types of IDs like barcodes. You might have heard about RFID and things like that. We project that, well, they're much smaller and cheaper to make. We project that the P-chips will eventually be linked together, millions or billions of objects through the internet. In terms of the biological applications, we have a whole suite of things that we do with multiplex assays, which I don't have time to go into, but it involves doing advanced diagnostics. But in this particular case, we realized that if we added high-speed sorting, we could do something called sequence-directed combinatorial chemistry. And what this is, combinatorial chemistry is taking a basic block, having lots of different possible things to attach to it, making every possible permutation. That's kind of what DNA is. In fact, in the early 1930s, when DNA was first discovered, people didn't think it could be used for the cells for information because there were only 4 different bases. They didn't realize how powerful it was. So what this means is that combining combinatorial chemistry with our P-chip and sequence-directed sequencing, we can make a genome in a single day. Okay, as long as we know that it's done. And here's how we do it. There are 2 key elements, a P-chip and a sorter. The P-chip will be reduced in size by 1,000-fold. That's easy to do today with the semiconductor techniques that are out there. And then we have a high-speed sorter. So what happens is we read the ideas based upon what the sequences are that we want. We sort them, we synthesize them, we pool. And this just keeps going around until you elongate the chain to what you want. You can do hundreds of thousands of chemical reactions together at the same time. The P-chips at this size, 1,000th the size of the current ones which I have here, for an entire human genome will occupy about 1 milliliter of volume. How are people doing it now? Well, there are some other ways out there with regular synthesis and microarrays and stuff like that. They're very, very tedious. They take a long time, and there's lots of errors. So when we look at what all this means is, you know, you might ask the question, we're going to go down to the basement and make mammals. No. There are a whole bunch of fields out here where right now people cannot make the nucleic acid materials they need in large enough quantities because of the limitations of expense. We project that our technology will be 3 to 4 orders of magnitude cheaper than the 2 techniques I showed you on the previous slide. So our X, in the solving for X, is a P-chip. We believe that we're going to have a lot of them, and it's, oh, what did I do there? So, yeah, let's just go to the last one. So we believe that the P-chip is the variable. Each one that we produce is unique, and it will be part of a much larger equation that will be in the toolbox that scientists and engineers have to improve the quality of products that are being delivered to us. Okay, thank you. Yes, so for example, if you sequence a gene and you say, I want to make variations of that in an experiment and see which one has a higher efficiency of, let's say, making a biological oil or something like that, you can create many different permutations and slight variations of the gene, splice them into organisms, and then see which one is more efficient.
So you're making it very cheap to write, not just read?
Well, the reading we don't do. That's being done by DNA sequencers, correct.
Are you worried about the moral implications and, you know, can be summarized in, say, the, you know, grey goo problem?
Well, there are ethical considerations, of course, and that's probably the subject of a whole separate lecture. And I was very careful to talk about the fact that we're not talking about higher forms of life here. So far, you know, the most publicity that has gone into this has been the formation of the first synthetic yeast that was done a couple of years ago. I don't believe that this will get to the point of a synthetic organism. I think what it is, its probably eventual targets are specific genes in chemical pathways that will be used for industrial production and things like that. It also turns out it's a very good way to design custom vaccines.
I think you mentioned up front that the current cost for sequencing the human genome is around $1,000. What do you see the P-chip in this technology getting that cost to in the foreseeable future?
Well, we're not sequencing again, we're synthesizing. Right now it costs about 10 cents a base, which means a letter, to synthesize, to add a base to DNA. We think it will be 3 to 4 orders of magnitude less than that, so that's about 1/100 of a cent.
So will this have an impact in customized medicine for each of us?
Absolutely. In other words, one of the visions, and this is not my vision, this is Craig Venter, who's the guy who's responsible for the Human Genome Project, has an idea of eventually having some type of pathogen that has been discovered, figuring out what the vaccine is for it, and emailing the sequence to the DNA synthesizer in Africa and making the vaccine on site.
Yeah, and if I understood you correctly, you're building a library of these plug-in kind of modules that could go into the...
Yes, for an entire genome, you'd have about 10 million fragments. And then there are tricks that you have in molecular biology that allow you to assemble the fragments into a linear structure in a very predictable way. There isn't time to go into how that's done. That's called cloning. But that's exactly right. And in fact, one of the implications of this is that we can very inexpensively make large biorepositories of libraries with different snippets of DNA that can be combined in different manners, in different ways. And it's not just for any one thing. It could be in plants, it could be in industrial enzymes, it could be vaccines, things like that, animal husbandry.
Yeah, I was thinking about vaccines.
A lot of people don't realize that even today about 4 or 5% of our gross national product is done with modified organisms, not just the stuff you hear about plants. But it's actually a significant part of the economy, and it's growing very, very rapidly. The problem is that the genetic community has not had the tools. This could be to the genetic community what the PC was to the computer world 20, 25 years ago.
So, as specifically as possible, describe for us at what stage you are with the technology. Okay,
well, the P-chips are done. Okay, I have some here. We have to reduce them to a smaller size. I already said that the semiconductor techniques are out there for the stuff. We've built sorters for the larger chips. They work fine. We have all the chemistry worked out. That's very well-known chemistry attaching it. It's just a question of taking all these different elements, most of which we have demonstrated at possibly a different scale, combining it and making sure the whole thing works.
What do you think the time horizon is for that work that you just described?
I'm going to refer to the inventor who's sitting here, Dr. Mandecki, to answer.
There will be different milestones, but the key milestone is 3 years.
That would be my guess, too. It's actually pretty close. Thank you.